Well, welcome everybody. My name is Dr. Fuz Rana. I'm a chemist. I'm also a Christian apologist, and I'm the Vice President of Research and Apologetics at an organization called Reasons to Believe. Reasons to Believe exists to demonstrate the scientific and, more broadly speaking, the intellectual credibility of the Christian faith and the Christian worldview, where we give primary emphasis on the scientific evidence for God's existence and the reliability of Scripture. If you want to know more about our organization, I would invite you to visit our website, www.reasons.org, where you can learn about the wonderful resources we have available for you. We are living through really unprecedented times, not only for our nation, but really for the world, where we're trying to battle this COVID-19 epidemic. And everybody is metaphorically locking arms together as Americans to try to do what we can to curtail and to kind of constrain the spread of the SARS-2 coronavirus. And as part of that, we are being asked to practice social distancing. We're asked to remain at a physical distance from one another. And in some states, like in the state of California, where I live in, the governor has issued a stay-at-home mandate where we're asked to remain in our homes and only leave our homes to go to the grocery store or to go to the pharmacy just to get essential items. And when we live through a time like this, where everybody's willing to do their part to try to bring this pandemic under control, it's very easy to become lonely, to feel isolated, to feel a lack of connection. And so what we want to do at Reasons to Believe is to take advantage of the technology that we have available to us to make sure that we are connecting with you, that we are doing what we can to provide you with not only new reasons to believe, but also providing you with reasons for hope. And I'm excited to say that we are launching a new program at Reasons to Believe called RTBLive. And the idea is that every Thursday at 1 p.m. Pacific Standard Time, 4 o'clock if you live in the Eastern time zone, one of the members of the scholar team will be broadcasting live a presentation where we will present to you reasons to embrace the Christian faith, where we will present to you either scientific or philosophical reasons to embrace the credibility of the Christian worldview, the Christian message. And I'm honored to be the person that's been invited to inaugurate these live broadcasts. And what I'm going to do today is to present a talk called Creating Life in the Lab. And I actually had the privilege of presenting this talk a couple of weeks ago, believe it or not. In Indianapolis. I was at the Indiana University School of Medicine. I was invited by the Christian Medical and Dental Association to present this talk to medical doctors and to medical students as part of an outreach effort, where I was tasked with the responsibility of showing scientific evidence for God's existence. And so I thought that this would be a great talk for me to share with you, since I literally just put this talk together to reach out to medical students, to medical doctors. And what we're going to be doing in this presentation is focusing on the work that's being done in an exciting area of the life sciences called synthetic biology. And synthetic biology is, again, one of the most interesting, exciting areas in the life sciences that really represents an amalgam of biology with engineering, where the goal of people working in this area is to create artificial, non-natural life forms. And as part of that endeavor, people are trying to create what they call protocells, cell-like entities that are assembled from simple chemical molecules, with the idea that these cell-like entities would begin to assume the properties of life. And so this is very exciting work. And the question on the table today is this: what does this work actually tell us about God's existence and God's role in the origin and the basic design of life? So that's what we're going to be taking on today. I'm going to be presenting for about 45 minutes, and then I'll open up the floor for a time of Q&A where you can ask me any question that you would like, not only on this topic, but also questions relating to any area of science and the Christian faith. Now, I can't think as a biochemist of any topic more fascinating than the origin of life question. And I first became interested in the origin of life question when I was a college student. In fact, it was the first day of my first biology class where I developed an interest in the origin of life question. Now, I was taking this class, Biology 101, taught by Dr. Bonnie Dean, to this day, one of the favorite professors that I've ever had. And I was taking the class in the summer between my senior year of high school and my freshman year of college. And the reason why I was taking Biology 101 as a summer school class before I even began college was because my father insisted that I do so. My father was a wonderful man, but he was a very strong personality, a domineering Indian father who wanted me to go to medical school. And when I graduated from high school, I really had no idea what I wanted to do with my life. And so my father insisted that I pursue medicine as a course of study and medicine as a career. And so I enrolled in a pre-med program at West Virginia State College at my father's urging. Sad to say, that was the sum total of the decision-making that went into my decision to pursue medicine as a course of study and as a career. So again, a bit embarrassed by that, but there you go. That's how I decided to become a pre-med student. But in the first day of our Biology 101 class, the first thing we talked about is this question, what is life? And of course, this makes a lot of sense because if you're studying biology, which is the study of living systems, you want to know what it is precisely that you are studying. And so I learned that very first day of class that biologists cannot define what life is. What they can do is they can describe what life is. They can delineate the universal properties that all living systems on Earth possess, but they can't actually define life. They can't produce a rigorous definition that will unequivocally distinguish living systems from inanimate systems. And I found that to be fascinating. It's been a perennial problem in biology, and that problem persists till this day. And so I wondered if I could be that person that could contribute to our understanding of what life actually is. And I realized, even as a young college student, that the two areas in the life sciences that would position me best to answer the question, what is life, is the study of biochemistry, which is the study of the molecular systems that constitute the cell. Because I figured that if we could understand the basic components of life and how they interacted, what their architecture is and their operation, that this would shed important insight into the question, what is life? And I also realized that the origin of life question related to the question of what is life as well. Because the origin of life, of course, is conceived usually in evolutionary terms, and it's the idea that evolutionary mechanisms essentially evolved simple molecules into ever-increasingly complex and integrated systems that eventually began to assume the properties of life. And so my thought was these two areas of study would give me the best ability to explain what is life. And I changed my career choice at that point in time. I decided I didn't want to go to medical school. I didn't want to become a pre-med student. I wanted to become a biochemist. And I wanted to pursue studies and a career in biochemistry. And so I ended up finding my calling in that first day of class in Biology 101, thanks to Dr. Bonnie Dean. Now, when it comes to understanding life's structure and operation at its most basic level, we've made incredible progress understanding the nature of biochemical systems. It's mind-boggling to think of what we learned in the last 35 years since I was a student entering into college. What we know about biochemical systems, as I think about this, is really staggering. And yet there's still, of course, much to learn, no question about it, but it's an incredible accomplishment as human beings to develop that kind of understanding of how life operates at a basic level. On the other hand, when it comes to the origin of life question, we've made virtually zero progress in the last 35 years. In fact, I think I would even argue that we really are no closer to understanding the origin of life today than we were 70 years ago or so, when Stanley Miller did his famous spark discharge experiments at the University of Chicago, experiments that launched and inaugurated the origin of life as a research discipline. It's not to say that there hasn't been a lot of work that's been done by origin of life researchers. Excuse me. There's been incredible work that's been done in origin of life research. Origin of life researchers are some of the most inventive, insightful scientists that I've ever had a chance to interact with. And there's a lot of ideas that have been proffered in the last 70 years. But in spite of all of this activity, we really are no closer to understanding the origin of life now than we were, again, when Stanley Miller did his famous experiment. Here's an interesting statement made by Paul Davies, a physicist and an astrobiologist, in his book, The Fifth Miracle. Excuse me. Now, Paul Davies says in the introduction of his book, The Fifth Miracle, which is a book on the origin of life, that when I set out to write this book, I was convinced that science was close to wrapping up the mystery of life's origin. Having spent a year or two researching the field, I am now of the opinion that there remains a huge gulf in our understanding. This gulf's not merely ignorance about certain technical details, it is a major conceptual lacuna. Now, Paul Davies is somebody that we want to pay attention to. Paul Davies is trained as a theoretical physicist and has written a number of popular level books on physics and on cosmology, where he not only presents lay accessible descriptions of the science, but begins to explore the interaction of science with ideas in philosophy and even in theology to some degree. And a few years ago, Davies developed interest in the origin of life question, and that interest culminated in a book called The Fifth Miracle. And after he published this book, he changed his career trajectory to now pursue work in astrobiology. But as a knowledgeable outsider, Davies basically acknowledged that the commonplace perception is that science is really close to figuring out how life originates. And maybe you share that perception. But the fact of the matter is, when you begin to dig into the details, you realize very quickly it is, again, a major conceptual lacuna. Now, as I mentioned, origin of life researchers have produced a lot of different ideas about how life could have originated. And these ideas are represented by a number of different chemical evolutionary models that can be grouped into three broad categories: replicator first scenarios, metabolism first scenarios, and membrane first scenarios. And for our purposes, we don't really need to know what these particular scenario groupings refer to specifically, other than to point out that there's been, again, a large number of models that have been proposed, three major groups of scenarios, and none of these approaches produce any kind of real understanding about the origin of life. Every approach to the origin of life leads to a dead end, results in essentially failure. For example, when it comes to replicator first scenarios, and again, there's a number of different replicator first scenarios that have been proposed, there are three major problems with these scenarios. what's called the monomer problem. How do you explain the production of the building block materials that you need to assemble information-rich biopolymers that could have the capacity for self-replication? There's ideas about how these monomers might form, but nobody really knows how they would form on the early Earth. There's the homochiral problem. These monomers have a chemical handedness, and they're either have a left-handed or a right-handed orientation with regard to the chemical groups in space, their arrangement in space. This is called the chirality of these molecules. And the chirality has to be identical for all of the subunits, and nobody knows how to attain a homochirality on the conditions of the early Earth. There's also called the homopolymer problem, where the information-rich polymers have to have a repeating backbone that's identical from subunit to subunit. And this is called a homopolymer. And if they don't have that repeating subunit, it's impossible to have a self-replicating system. A few years ago, I had the privilege of attending an Origin of Life conference in Oaxaca, Mexico, and the plenary talk at that conference was given by Leslie Orgel, when he was alive, one of the world's preeminent Origin of Life researchers. And Orgel in the 1980s proposed the RNA world hypothesis, which is the leading replicator first scenario. And he was asked to give a summary of where we were at with regard to the RNA world model and replicator first scenarios in general. And in response to that, he concluded his lecture where he delineated the types of problems that I've summarized here with this statement: it would be a miracle if a strand of RNA ever appeared on the primitive Earth. Now, to be fair, Orgel was deeply committed to a chemical evolutionary approach to the origin of life, but he was pointing out, in a sense, the very real, what appears to be insurmountable obstacles to explaining the origin of life through replicator first scenarios. Metabolism first scenarios, too, are riddled with problems. And for example, metabolism first scenarios would involve the emergence of what are called autocatalytic chemical cycles, where you have essentially chemical materials reacting with one another and forming essentially pathways that would cyclize, forming again a catalytic cycle, where one person or one molecule in that cycle would be interconverted to another molecule. And these kind of autocatalytic cycles are highly susceptible to chemical interference, and it's difficult to envision them existing on the early Earth. The chemical reactions needed to support these autocatalytic cycles are way too slow, and they require mineral catalysts that have a very limited capacity for specificity when it comes to catalyzing these reactions. Orgel, in response to these problems, wrote a critical review of metabolism first scenarios, where he argued that these would require an appeal to magic, a series of remarkable coincidences, a near miracle. A few years ago, I collaborated with another chemist by the name of Jackie Thomas, and she and I published a paper in Origins of Life and Evolution of Biospheres, where we pointed out the intractable problems associated with membrane first scenarios for the origin of life. So the whole point is this, that every approach that's taken to try to explain the origin of life, again, leads to dead end, leads to frustration. We do not understand how life originates. And this is where synthetic biology enters into the picture, because many origin of life researchers are turning to synthetic biology with the idea that this area of biology could very well represent a fresh, insightful approach to the origin of life problem that might help us to overcome these intractable problems that we currently are facing with all the different chemical evolutionary scenarios. Now, as I mentioned, synthetic biology is really an amalgam of biology and engineering, where the goal is to create artificial non-natural life forms. And there's two broad approaches that are being taken. One is to re-engineer existing organisms so in such a way that they no longer represent organisms that you would naturally find in the world of nature, that these would be artificial organisms that were re-engineered from existing organisms in nature. The other approach is to start with simple chemical materials and try to assemble them into protocellular entities that would be essentially life forms of our own creation from the bottom up. Now, the reason why people want to do this is they see synthetic biology as the pathway to the next technological revolution, with the idea being that we could essentially create these artificial organisms to serve as bioreactors, where we could feed these organisms relatively inexpensive renewable materials from the environment, and using renewable sources of energy like sunlight, coax these organisms into converting these raw materials into highly valuable products, medicines, biofuels, a wide range of industrial products. People even envision these artificial organisms as playing a role in agriculture, helping us to feed the world, helping us to provide food resources for an ever-growing population around the world. But there are a number of people who are interested in synthetic biology that realize that synthetic biology has value that goes beyond merely its technological utility. That because we are trying to create artificial cells in the lab or re-engineer new life forms, we are in effect taking control of evolution ourselves. We are essentially directing the evolutionary process, so to speak, to create these new novel life forms. And so the thought is that this approach could actually give us insight into the origin of life, into the evolutionary history of life. And it kind of is in the vein of a statement made by the late Nobel laureate Richard Feynman, the physicist who said that if I can build it, it means that I understand it. And people are taking that idea and they're using it to try to, again, understand how life could have originated through some kind of evolutionary process, with the notion that if we can build life, that means we should understand it enough, to know how it would have evolved. Now, towards that end, when you look at work that's being done in synthetic biology, it is essentially a mirror image of the work that's being done by origin of life researchers. In synthetic biology, scientists are going in the lab and they're trying to take simple molecules and assemble them into these ever increasingly complex chemical super systems, where the chemical systems are integrated with the eye towards trying to create, again, artificial protocells that have the properties of life. Origin of life researchers trying to understand how life evolved on Earth are trying to go in the lab and recapitulate the different steps that they think took place in the origin of life, starting with simple chemicals and increasingly augmenting their complexity to the point that they now have, again, these protocellular entities that are steps away from the very first life form. They both are doing the same work. The difference is origin of life researchers are constrained by the resources that would be available to them on the early Earth and the conditions of the early Earth, whereas synthetic biologists are free to explore all kinds of options, thinking outside the box. And this is why origin of life researchers think that synthetic biology may provide that insight that could help explain how life could have emerged through evolutionary means, because of the creativity associated inherently with work in synthetic biology. But it's interesting to me that today, synthetic biology and origin of life research is beginning to meld quite a bit, where some of the most successful workers in synthetic biology are actually origin of life researchers. And some of the most interesting work in the origin of life is actually being done under the auspices of synthetic biology. And so it's in light of this that I want to ask the question, what does synthetic biology tell us about the origin of life? What does synthetic biology tell us about the origin of life? And to answer that question, I'm going to talk about a study done by a team of Japanese researchers where they created self-replicating protocells. And this is going to be an exemplary study. And it's exemplary not only in the high quality of scientific work associated with the study, but it is exemplary in it that it is a representative study that serves as an ideal case study that allows us to extract principles about the origin of life from, again, work that's being done in the attempt to create artificial protocells. And what this research team did is they created protocells that could grow and replicate in the laboratory for multiple generations. That's an amazing accomplishment. But what's even more astounding is that they were able to couple the growth and replication of the protocells with the replication of DNA in the interior of the protocell. And on top of that, they figured out a way to replenish the nutrients in the protocell after the growth and division process took place, allowing the protocells to, again, replicate over and over and over again for multiple generations. So what I'd like to do now is describe in detail what these researchers have done. Now, for some of you watching this, these next few minutes are going to be on the technical side, and you're going to feel as if maybe this stuff is over your head. Please bear with me, because after we talk about some of the technical details, we'll talk about the big picture ideas, and you should be able to follow along very easily at that point. For others of you, you're going to be frustrated because you're going to feel as if I'm not technical enough. So just please bear with me. Hopefully we'll hit a happy medium as we go through this description. So now this slide shows a summary of what these researchers did. Is they created these protocells that had encapsulated within their interior a double-stranded piece of DNA, a small piece of DNA called a primer, an enzyme called DNA polymerase, which replicates DNA, and then the building blocks for DNA. And they were able, through a thermal cycling procedure, to essentially stimulate the DNA to replicate in the interior. They then added a compound that would incorporate into the vesicle walls of the protocells, the walls of the protocells, which would cause those protocells to grow and then eventually become unstable and fissure into two daughter cells. And then they added what they called conveyor vesicles loaded with nutrients that could replenish the depleted nutrients in the protocell. And this involved these conveyor vesicles associating with the surface of the protocell, and then they would alter the pH of the solution, causing a fusion, and voila, they wound up with a daughter vesicle, a daughter protocell that was identical to the parent, and they could repeat this process again and again and again. Here's another slide that gives you some sense for what it took now to pull this off. And in order to build the vesicles, the protocells, they had to very carefully select the just right phospholipid components that had the just right chemical and physical properties so that the vesicles would form. They had to carefully design two non-natural synthetic lipids, one called the membrane precursor, one called the amphiphilic catalyst, and that amphiphilic catalyst would react with the precursor, the membrane precursor, to produce the membrane molecule and an electrolyte. And we'll explain what these non-natural synthetic lipids were doing in a minute. But the point is that they had to very carefully select the right types of phospholipids in order to form the two different types of vesicles, the protocells and the conveyor vesicles. And they had to design and synthesize two non-natural lipids in order to couple DNA replication to the growth and division of the protocells. This slide gives us, again, an overview of what these researchers did, where, again, they, first of all, formed the vesicles by adding the just right phospholipid with the amphiphilic catalyst, compound C, if you will, in the right composition to form the vesicle walls. They had to do a process where they added to solution the nucleotides, the DNA primer, the DNA polymerase. They had to dry the system down, reconstitute it, dry it down, reconstitute it again, in order to get the encapsulation to take place. Then to trigger the replication process, they had to very carefully ramp up the temperature, which would cause the DNA double helix to unwind, exposing single-stranded DNA. The DNA primer would interact with the DNA polymerase, and the DNA polymerase could then read the single strands of DNA, assembling the daughter DNA strands, and voila, DNA replication took place. And then they had to very carefully cool the system back down so the DNA double strands would re-anneal. They then added the membrane precursor, which would incorporate into the membrane. But it turns out that in the membrane, that amphiphilic catalyst would react with it to produce the membrane molecule, and then it would produce an electrolyte which would diffuse away from the protocells. And this is critical because it thermodynamically drives the reaction in the forward direction. They then added the conveyor vesicles, and the conveyor vesicles were made up of phospholipids so that one of them had a negative charge, meaning the conveyor vesicles had a surface charge that was negative. The protocells have a surface charge that's positive because the amphiphilic catalyst was actually positively charged. This allowed for the vesicles to associate, and then by altering the pH, it caused the fusion of the vesicles in order to complete the replication process. This next slide shows again the same experimental procedure, but now we're going to focus on what was going on in the vesicle wall, in the interior of the vesicle wall. Because compound C is positively charged, when DNA would complete the replication process, because it's negatively charged, it would in effect associate with the vesicle wall. And this is how they were able then to couple a DNA replication to the growth in the division of these vesicles. Okay, so now we are done with the real technical part of the presentation, but the researchers who did this work, which again is exemplary work, this is science at its very best. As a biochemist, I marvel at what these researchers have done. These researchers conclude their paper by arguing that this work demonstrates how collaborative dynamics emerged under certain circumstances. In other words, what these researchers are arguing is that in order for a self-replicating protocell to emerge, you literally have to construct independent biochemical systems, and then you have to figure out ways for those systems to collaboratively interact in order to sustain the properties characteristic of living systems. And these researchers feel that this work provides valuable insight that is going to pave the way for how chemical evolution could have transpired. And it's interesting to me that many people that work in origin of life research argue that these kinds of studies, again, give us critical insight into chemical evolution, what's required for chemical evolution to transpire. It elaborates principles that apply to our understanding of the chemical evolutionary process. And many origin of life researchers and synthetic biologists will argue that if we can actually create protocells in the lab, it means that it's all that much more reasonable that chemical evolution could take place on the early Earth. That these kind of experiments give researchers hope that somehow the origin of life could happen through chemical evolution. In fact, there are some people who look at this work and argue that not only does this work provide scientific insight, but it actually offers us philosophical and even theological insight into the nature of life itself. One of these is philosopher Mark Badeau, who is the chief operating officer for a company called ProtoLife. And in an interview, Badeau said this, that creating protocells has the potential to shed new light on our place in the universe. This will remove one of the fundamental mysteries about creation, in the universe and our role. We are doing things which were thought to be the province in some quarters of God, like making new forms of life. In other words, people see this work in synthetic biology and the creation of protocells as essentially eliminating any need or necessity for a creator at all. But here's what I think is ironic about this work, and that is, I think instead of demonstrating that chemical evolution is a viable approach to explaining the origin of life, I think ironically work in synthetic biology does the exact opposite of what Marc Bedeau claims. I think it actually demonstrates the necessary role for intelligent agency in transforming into simple molecules into the very first cells. That this provides us with an empirical set of evidence that a creator must have played a role in the origin and the basic design of life. Because when we look again at the study done by this team of Japanese researchers, these are some of the best minds in the world who are relying on hundreds of years of accumulated knowledge to develop highly elaborate, sophisticated strategies that they then implement through detailed laboratory protocols that require highly skilled, highly knowledgeable scientists working under very precise controlled conditions to execute. The intelligent agents are playing a vital and central role in the conversion of molecules into protocellular entities. And so therefore, by analogy, we would expect that an intelligent agent, a creator, must have played a role in the origin and the basic design of life. These researchers had to very carefully choose the just right phospholipids with the just right properties to form the vesicles. They had to synthesize two non-natural lipids with the just right set of chemical and physical properties in order to couple the growth and the replication of the vesicles with DNA replication in the vesicle interior. They had to very carefully adjust the composition of the different lipid components in the vesicles in order for this system to function. They had to very carefully control the conditions in order to encapsulate the materials in the vesicles to begin with. They had to very carefully develop and control the temperature changes in order to trigger DNA replication and its re-annealing process. They had to very carefully add the just right amount of lipids to destabilize the vesicles, and they had to very carefully control the conditions when they added the conveyor vesicles in order to replenish the daughter protocells. In other words, these researchers, again, functioned as the intelligent agency that was critical to pull this off. These researchers make this statement towards the conclusion of their paper. To achieve this goal, we selected well-defined, suitable lipids and macromolecules, including newly designed ones, and constructed a giant vesicle-based model protocell that links self-replication of information molecules with the self-reproduction of a compartment. These researchers can't get away from design language. They have no choice but to employ design language because they were functioning as intelligent designers. Now, this conclusion that we derive from this particular study would apply to any study that's done in synthetic biology. This, again, is just a case study, just a representative study, but we could go through every study in synthetic biology designed to create protocells and essentially analyze it in the same way I analyze this study and arrive at the same conclusion. Intelligent agents are necessary for the origin of life. Now, let's go back to this slide that we showed earlier. Because if the conclusion from synthetic biology is that intelligent agency is necessary for the origin of life, then what is the conclusion that we derive from work in prebiotic chemistry? We've already talked about the fact that we don't know how life originated through chemical evolution, but when we examine in detail the work that's being done in prebiotic chemistry, what conclusion can we draw from that work? Do we draw a conclusion that is comparable to the one we drew from the studies in synthetic biology? Well, people that are working in prebiotic chemistry, even though they don't know how life originates, feel comfortable, feel confident that chemical evolution is the way to explain the origin of life. And this is because that in a laboratory, they've identified chemical and physical processes that in principle could contribute to chemical evolution. They've identified chemical reactions that can make the building block materials for life. They've identified pathways to assemble those building block materials into biopolymers. They can evolve functional biopolymers in a laboratory that can carry out a wide range of biochemical activities. They can generate self-replicating systems and manufacture protocells, as we've talked about. And many origin of life researchers will look at this and say, yes, we agree, we don't know how life originates, but these studies represent clues to the origin of life, clues to how chemical evolution transpired, and give us confidence that chemical evolution could indeed explain the origin of life. But the problem here is that the success that they've achieved in the laboratory is a false success. Because in the laboratory, just like the work involved in synthetic biology, you have brilliant organic chemists, some of the best minds in the world, who are designing elaborate experimental procedures and then carrying them out under very carefully controlled conditions in the laboratory, where they very carefully assemble these experimental apparati, where they add the just right solvent with the just right pH and temperature, the just right ionic strength. They add the just right chemicals at the just right time and the just right concentrations. They're working with chemically pristine conditions where there's no interfering molecules. And under those conditions, they have success. But the question is, does this work in the lab translate to the conditions of the early Earth? And in my book, Creating Life in the Lab, published in 2011, I argued that the answer to that question is no, that the conditions of the early Earth are uncontrolled. They're chemically and physically complex conditions. It's very difficult to envision how the laboratory controls would ever have any relevance to the uncontrolled conditions of the early Earth. And what's interesting to me is that in recent years, origins of life researchers are now acknowledging this problem that I identified in my book, Creating Life in the Lab. A German chemist who works in origin of life research about a year ago published a paper in Nature Communications where he said this: it's not easy to see what replaced the flask, pipettes, and stir bars of a chemistry lab during prebiotic evolution, let alone the hands of the chemist who performed the manipulations. And yes, most of us are not comfortable with the idea of divine intervention. Let me illustrate this problem very quickly with the RNA world scenario. This was the idea that was proposed by Leslie Orgel in the 1980s, an idea in which he said it would be a miracle if a strand of RNA ever appeared on the primitive Earth. This is a leading explanation in the view of many people for the origin of life. And the idea here is that the very first biochemistry was based around RNA, and then later this RNA world evolved to give rise to the DNA protein world. And there's a number of reasons why people think the RNA world is a valid idea. I'm not going to get into those other than to talk about a study that was initially done in the 1990s, and then there were a number of follow-up studies that were done all the way into the mid-2000s, where researchers showed that you could assemble RNA polymers on clay surfaces starting with RNA building blocks. Now, this was considered to be significant in terms of the origin of life research program. It was considered to be a major step forward in validating the RNA world, because people envisioned that there would be minerals on the early Earth. And minerals like clay, which have these interstitial spaces between layers of the clay, could accumulate building block materials at very high concentrations. It would be an environment largely devoid of water, and that the metals in the clay could serve as catalysts driving the production of RNA polymers. And so people were really excited about these results. But when you examine these studies, you see that these studies are only successful because of the role the research is playing. These experiments are done under chemically pristine conditions. They have to be done in distilled water. You cannot have materials present in the system that likely would be present on the early Earth at high levels, because these either interfere with the growth of the RNA chain or cause the RNA chains to break down once they form. The researchers stop the reaction at the just right time before the RNA polymer gets too long and becomes irreversibly absorbed onto the clay surface. Because if this happened on the early Earth, that molecule would no longer be available to participate in the origin of life process. It would frustrate the origin of life process. And then finally, in order to perform these experiments, you actually have to buy the clay from a particular supplier and then treat the clay in a very special way in the lab in order for it to function as a catalyst. And on top of that, you have to use not the building block materials, but chemically modified building blocks that are poised to be more reactive. As Paul Davies says, as far as biochemists can see, it is a long and difficult road to produce efficient RNA replicators from scratch. This conclusion has to be that without a trained organic chemist on hand to supervise, nature would be struggling to make RNA from a dilute soup under any plausible prebiotic conditions. Simon Conway Morris, an evolutionary biologist, goes one step further when he says this: prebiotic experiments designed to explain one or other step in the origin of life are either of tenuous relevance to any believable prebiotic setting or involve an experimental rig in which the hand of the researcher becomes, for all intents and purposes, the hand of God. In other words, what we see is that when we analyze the work in prebiotic chemistry, we arrive at the same conclusion that we arrive at when we evaluate the work in synthetic biology, that we actually see in both instances the critical role intelligent agency must play in the origin and the design of life. That this is a very powerful empirical argument for God's existence, where it's an argument based empirically on what we observe time and time again when people step in the lab and try to create protocells, either under the auspices of synthetic biology or under the auspices of prebiotic chemistry. So the question then becomes, very quickly as I bring everything to a close, why is it that origin of life researchers are reluctant to embrace the role of intelligent agency in the origin of life? Well, this statement by Paul Davies, I think, is rather revealing towards that end, where he says, although biogenesis strikes many as virtually miraculous, the starting point of any scientific investigation must be the assumption that life emerged naturally. In other words, it's the philosophy of science that is the problem, not the scientific evidence and what that evidence is actually telling us. Because the way science is constructed today, the way it operates, is under the auspices of methodological naturalism. This is the organizing philosophical framework for science, which in a nutshell says that when you investigate nature, phenomena within nature, your explanations are limited to mechanistic natural process explanations. The divine cannot be evoked. The supernatural cannot be evoked. And so it's for philosophical reasons that people are committed to a chemical evolutionary explanation for the origin of life. It's not because the evidence supports chemical evolution. In fact, the opposite is the case. The evidence, if we're willing to be open-minded, actually supports the necessary role of a creator in the origin and design of life. And so this quest that I was on when I was an undergraduate student that began about 35 years ago is essentially arrived at its destination, where I think we actually do understand, again, life, what life is, and it is the product of a creator, that life is the product of a creator, and that if that indeed is the case, it means that there must be an underlying meaning and purpose for life here on Earth, which means there's likely an underlying meaning and purpose for our lives as human beings. And once we discover that there is a creator that has played a role in the origin and the design of life, I think it's the next question is, who is that creator? Do I relate to that creator, and how? And I think that the answer to all of these questions are found in the gospel of Jesus Christ. So I'm going to go ahead now and end the formal presentation and really open up the floor to you to ask your questions. And I can see that there's already a question coming in. So let me see if I can figure out how to stop sharing my screen. And while I do that, I'm going to take this question here from Keenan, who says, doesn't the emergence of a new mutated virus that are able to live better in their atmosphere show that macroevolution can happen? Okay, so great question, Keenan. And I would respond to that question by saying that when you think about evolution, it's really important to think about evolution in the appropriate way. Because when you talk about evolution as a concept, it essentially is referring to change over time. And when you apply that concept to biological systems, that change over time can happen on a number of different scales. And so I like to think about different types of biological evolution. So for example, what we are talking about today is known as chemical evolution, which is describing the origin of life. But you also have what I would call microevolution, variation happening within a species. There's also speciation, where one species give rise to another species. You also see evolution happening with microorganisms, and then evolution happening at the macro scale, where one major group is evolving into another major group, where evolutionary processes are creating genuine biological novelty, genuine biological innovation. And to me, I think that it's very clear that when it comes to chemical evolution, when it comes to the origin of biological novelty, that the evolutionary paradigm lacks robust, vigorous explanations. When it comes to microevolution, speciation, and microbial evolution, those areas of evolution are well established. They're non-controversial. So just because a virus is able to mutate to form a new type of virus that now hosts and infects human beings doesn't mean that the evolutionary paradigm in its entirety is valid, nor does it mean macroevolution is valid. Microbial evolution is a completely distinct type of evolutionary process compared to macroevolution. All right. All right. Okay, here's a question from Karina. Can circular polarization on the early Earth solve the monomer problem? Thanks for the great presentation, Fuzz. Can you stop sharing your screen? Yes, I would love to be able to stop sharing my screen. This is what we call in the biz a technical glitch here, technical problems. Can't quite figure out how to stop sharing my screen, so I apologize for that. But yeah, one idea to explain the origin of homochirality is through the use of circularly polarized light that would essentially impinge upon monomers, and in the process essentially destroy preferentially one monomer of a particular chirality while preserving the monomers that have the opposite chirality. And the problem with this is that the destruction is never quite that clean to begin with. And even if it was clean, that process of exposing these monomers to circularly polarized light will not only destroy the molecule that you want to destroy, but also destroy some of the other molecule as well. And you have to have a very highly intense circularly polarized light source to completely destroy the monomer of one chirality. And in the process, you're going to wind up, in a sense, destroying all the materials that would be present. And under more realistic conditions where the energy levels are more moderate with the circularly polarized light, the best you can usually attain is a few percentage of chiral enrichment. You never get full 100% chiral enrichment. Here's a question from Kirby. Have any of the researchers been able to create information which might be the basis for RNA, DNA, or proteins? The answer to that question is yes. I think actually researchers are able to create information in prebiotic simulation experiments. And I know that this answer is contrary to what many people have often heard, because it's quite common for Christian apologists to make the claim that evolutionary mechanisms cannot generate information. And that's simply not true. You can assemble RNA building blocks in a laboratory, again, as we described, and you can start with those RNA building blocks which have a random sequence of RNA, and then through a process in the laboratory called in vitro evolution, actually evolve those RNA molecules to be information-rich molecules that produce RNA molecules that can carry out specific types of catalytic processes. But the problem with those studies is essentially the same kind of problem that we've talked about in the presentation. That is, in principle, evolutionary mechanisms can generate information starting with random nucleotide sequences. But in order to do that, you have researchers that are intervening in a critical and necessary way to manipulate the evolutionary process so that that information is generated. So it's the same kind of critique that we applied to the self-replicating protocells or to the assembly of RNA building blocks into RNA polymers on clay surfaces. Here's a question from Sploden. How can you tell the difference between something that is designed and something that is not designed? Oh, that's a great question. And sorry, give me a second here. I'm having some technical problems on my end. So how can you tell the difference, Sploden asks, between something that is designed and something that is not designed? I've written a number of articles that are available on our website at reasons.org, or if you go to my blog, The Cells Design, you'll see these articles where I make the case that there's a number of different scientific disciplines that actually are able to detect design in nature, in the natural realm. SETI, the Search for Extraterrestrial Intelligence, is an intelligent design research program where people claim that you can distinguish between electromagnetic radiation emanating from a natural source from electromagnetic radiation emanating from an advanced intelligent civilization. Or you have anthropologists who can distinguish between a rock shaped by natural processes and one shaped by an intelligent agent, by a hominin of some sort. And what I've done in these articles is looked at what was the criteria that these researchers were using to distinguish between something that was designed and something that was produced through natural processes. And so, for example, Seth Shostak at SETI argues that the criteria that is used is artificiality, where it is essentially a feature that you would not expect to result from natural processes. Or if you look at the work done by archaeologists, what they'll do is, again, they'll note that a particular feature of that artifact or where that artifact is found in the archaeological site displays artificiality. And then in both the case of SETI or in anthropology, what you then do is look to see, are there natural process mechanisms that you may have overlooked that could reasonably account for those processes? And again, if you can't, then the most reasonable conclusion is that these processes or these objects must be the product of a designer. And then also something else that you see taking place in archaeology is that researchers actually go into the lab and they will try to replicate that particular archaeological artifact. Again, with the idea of trying to understand how that artifact was produced, but also what it would take in terms of cognitive ability and manipulative skill to create the artifact. But in doing so, they again are contributing to the evidence that that artifact was designed by illustrating what does it take to produce that artifact. And so in a sense, what you have are really three criteria. Does it appear to be artificial? Does it, again, defy some kind of mechanistic explanation? And then what does it take to create that system? And so when it comes to biochemical systems, what we talked about are two of those criteria today. That the fact that we don't know through natural process mechanisms how life originates, and when we try to create life in the lab, it's very clear intelligent agency must play a role. And in my book, The Cell's Design, I present essentially a case for design of biochemical systems by looking at the features that they possess that display an artificial quality that allows us to conclude that they're designed and in the process reinvigorate the watchmaker argument of William Paley. Okay, this is a question from Chon. Oh, say Chon Ho Yu. Okay, can you address God of the gaps? Someone may say, even if you cannot explain the origin of life by science, you should not evoke God. Well, I'm very sympathetic to that criticism of a lot of design arguments or arguments for the role of a creator, because many times what people do is they claim, well, if evolution can't explain it, or if we currently don't know the evolutionary explanation, then it means that there must be a mind that's behind this. And kind of the reasoning that undergirds that kind of argument is that you have two options. Either it's evolved or it's designed. Well, if we can conclude it's not evolved, therefore it must be designed. You're kind of backing into the argument for design using that kind of reasoning, where it's really an indirect argument for design based on what evolution can or can't do. But notice that what we argued here today wasn't that at all. What we argued here today was, yes, indeed, evolutionary mechanisms can't explain the origin of life, but then we went one step further and said, okay, what does it take to create life in a laboratory setting? What does it take when we try to recapitulate chemical evolution? And what we see empirically is that intelligent agency is required. So this is not a God of the gaps argument, but rather it's essentially an argument from what the empirical data says. It's a conclusion that we're drawing positively from what the empirical data tells us, which is intelligent agency plays a necessary and vital role. in these kind of transformations. Or if you're looking at certain features of biochemical systems that, again, you are arguing display artificiality, then again, that artificiality in and of itself is a positive piece of evidence for a creator. It's not a God of the gaps argument, but rather it's an argument based on the attributes that particular system possesses. And so I would say that I agree. We don't want to argue God of the gaps. We want to argue for a creator based on positive evidence for that creator's role, in this case, in the origin and the design of life. And I think the part of the problem that many people have with evoking a God of the gaps explanation, or check that, with evoking a creator to explain, again, some feature in nature and why they label it as God of the gaps, actually has nothing to do with the fact that it's a God of the gaps argument, but rather it has to do with the fact that there's a violation of methodological naturalism. And so they argue that to conclude a creator means that you are implicitly violating methodological naturalism. And because we can't do that, we're going to label that our discomfort as God of the gaps, when in fact the discomfort has nothing to do with it being a God of the gaps argument, but rather violating methodological naturalism. Okay, here's another question. Let's see, can the building blocks of life naturally coexist? Lipids, carbohydrates, amino acids. Yeah, these building blocks can, yes, indeed, naturally coexist. But if what you're getting at with that question is, can they form under the same sets of conditions on the early Earth? That question is no. Many times the sets of conditions that you need in order to form one type of building block material would actually be the sets of conditions that actually would destabilize and cause the degradation of other types of building block materials, or would inhibit their formation to begin with. And in fact, this was something that Jackie Thomas and I pointed out in our paper that we published in Origins of Life and Evolution of Biospheres, is that when you look at the sequence of events required to explain the origin of life through a membrane first scenario, the conditions required to effectively execute one step essentially make the second step impossible. Would be conditions that would destabilize or inhibit or frustrate the second step or the third step in the process. Okay, here's a question from Ernie Jerome. Way back at step 1, where did the initial RNA come from? I'm not sure what you mean here by step 1 specifically, but let's say we go back to the RNA world hypothesis. And as I mentioned, in the RNA world hypothesis, the problems that are associated with it are the monomer problem, the homochiral problem, and the homopolymer problem. And each one of those problems represents, in effect, the different steps towards the RNA world. And so the first thing you'd have to do is generate the RNA building blocks on Earth. And it's right now, there's no chemical route that's been identified in the laboratory that would actually be productive under the conditions of the early Earth. And there's a number of different approaches that people have tried to create the different subunits of ribonucleotides, the nucleobases, the sugars, the ribose sugar, the phosphate, and then to assemble them into ribonucleotides in and of itself is a challenge. There's been some rather inventive work by the Sutherland group in the UK, where they've come up with a 5-step reaction that could generate ribonucleotides. But again, it's questionable if that reaction could work under the conditions of the early Earth. So it's a real struggle to even get the RNA building blocks. But even if you assume that there was some way to get the RNA building blocks, as I point out, to assemble them into an RNA polymer is a very challenging step. But even if you could assemble them into RNA polymers, to get them then to evolve into a wide range of RNA polymers that carry out wide range of biochemical activities is possible in the laboratory using in vitro evolution, but there's no way to envision how that process would work under the conditions of the early Earth. So at every step in the process, you are stymied. So your point is, how do you get the RNA to begin with is a legitimate question, but it's even a better question to say, how do you get the building blocks to begin with? That's probably even a more profound question and just as big of a challenge as how do you get the RNA to begin with? So your point behind the question is a good point, if I understand what you mean. Here's a question from Maria. Would an artificial cell be representative of a natural living system if it could only replicate in isolation with no visible purpose, or is evolutionary purpose or functionality necessary? Well, you know, you're asking a question that really is insightful and is really profound and really gets to the heart of the comment that I made at the beginning of my presentation, which is we really don't know how to define life. We can describe, again, the properties that all living systems universally have, but we cannot actually define what a living system is. And so when we create a protocell, as in an attempt for that protocell to be some kind of representative of a natural living system, it's going to be rather arbitrary as to whether or not we actually conclude that it truly is a representative of a natural living system, or is it just simply a very sophisticated chemical super system that can do some interesting things that are reminiscent of life. So without a proper definition of life, it really is difficult to know what we actually have created when we're creating these protocellular entities in the lab. And so I would argue that these systems that we're creating, as sophisticated as they are a far cry from the minimal complexity that's required for even the simplest conceivable cell. And so I don't think we really are truly creating facsimiles of a true cell. We're not really truly creating representatives of a natural living system whatsoever in these experiments, to be certain. But there will be a time in the future where people will have declared that they actually have created a living system, but it's going to be really, again, dependent upon what you mean precisely by life. But over time, people are going to create protocells that will, again, increasingly have all the different attributes, or a growing number of attributes, that would be shared with living systems, with true cells. And at that point, again, people will try to make the argument that what you have there is a life form, when, again, I think there's a legitimate debate to be had whether that will actually be the case whatsoever. But when you look at the complexity of even a minimal cell, it's far more complex than what we're looking at here. The complexity of a minimal cell is absolutely staggering. And there's no prospects of replicating something like that using the bottom-up approach in the near future by any means. Okay, looks like we're about to wind down here. I'm going to try to take maybe a couple more questions. The last question here, actually, I'm going to take one more question. What is the ultimate mechanism that drives the evolutionary process? And is this even knowable? Well, I mean, the evolutionary biologists would say that the ultimate mechanism is variation of the genetic material caused by mutational events that then is operated on by some form of selection, whether it's natural selection or sexual selection of some types. Something that takes those systems that are more persistent in the environment and have the capacity to replicate with the greatest degree of fidelity and with the greatest degree of speed, are going to be those systems that are going to ultimately persist in the environment. So I think people do understand what the nature of the evolutionary process is. The question really on the table is, can that process actually explain not so much the diversity of life on Earth, but can it explain the innovations that we see throughout life's history. And I think that's a place where that mechanism seemingly falls short based on everything that I've seen that mechanism being capable of doing. It does a wonderful job of explaining variation within a species or accounting for speciation. It does a wonderful job of explaining evolution in microorganisms, but it doesn't do a great job of explaining biological innovation. Okay, it looks like we're probably close to wrapping up, if I'm not mistaken. I'm supposed to give you a web address if you want to get something from us, but I seem to have lost that web address. Let's see. Oh, here we go. There is actually a link in the description, in the description of this YouTube video that you're going to be watching, or that you have watched and that you will be watching if you're watching the recording. And that if you actually click on that link, it'll take you to a web page where you can give us information, and in exchange, we will send you a PDF a copy of the slides that I used in my presentation and other free resources as well. So you want to click on that link to take advantage of some free resources that we have for you. And if, again, you provide us with your contact information, we'll be able to keep you abreast of the latest resources that we have available at Reasons to Believe, many of them at no cost to you, that will help you to see the scientific evidence for God's existence. Okay. All right, so I'm instructed that our time is up. And so I just want to say thank you for joining us in this inaugural broadcast of RTBLive. I apologize for some of the technical glitches that we had during the Q&A session with the backdrop, the screen background. That's exclusively my fault, my problem. We have a wonderful team at RTB that has done a great job of helping us to put these broadcasts together. I just invite you to stay tuned for next week. There's going to be another RTBLive available to you at 1 p.m. Pacific Standard Time, 4 p.m. if you're in the Eastern Time Zone. You can catch the live broadcast, or you can also catch the video recording of those broadcasts later on our YouTube channel. Reasons to Believe 1 is our YouTube channel. But until then, I just want to say thank you again so much for watching this broadcast. Please stay safe, stay healthy in this very difficult and challenging time. Don't lose hope, don't despair, because remember, the more that we learn about science, the more that we have reasons to believe and reasons to hope. So God bless you. I will see you next time.